Hi everyone, myself Abhishek Sharma. I'm from National Center for Biological Science, Bangalore, India. I have a joint student between Professor Sudhavni and Professor Sudhir Krishna. Today I will talk about my work on dengue NS1 protein, where I have carried out bioinformatics analysis to shed the light on its evolution as well as implication in identification of druggable as well as functional sites. NS1 protein is also called as antigenic protein, highly conserved in nature. It is present in three states, which is monomer, dimer, and hexamer. It translates as monomer, immediately form dimer, and in the Golgi operators, this dimeric unit interact with other three dimeric units to form a hexameric state. It is very critical protein as it is shown to interact with the dengue as well as host intact proteins, as well as critical for viral replication. The secreted NS1 protein is important for dengue pathogenesis as it inhibits complement pathway, it activates monocytes, it antibodies which are generated against this soluble NS1 protein are also act as autoantibodies, make host more susceptible, and in the severe conditions, it resulted into dengue hemorrhagic fever as well as shock syndromes. Also, it activates endothelial cells, which is the primary cause for causing cytokine storm. So with this, the NS1 is highly important protein for dengue replication as well as its pathogenesis. So we ask why NS1 is highly conserved despite of having evolutionary pressure. If you see other highly immunogenic protein such as envelope protein, spike protein in the case of SARS, they are highly diverse in nature. So what restricting NS1 protein? As you can see, it is seeing similar kind of immune pressure. So we hypothesize that this quaternary arrangement of NS1 protein from monomer to dimer to hexamer are creating some sites which are mutationally inactive, but in the case of linear sequence can accommodate mutations. So this gradual increase in the states can decrease the ability of accommodate in mutations. So how to check it? Firstly, we need to have a structure to start with this kind of hypothesis. So we take the already solved structure from PDB 406B. It has some missing loop regions. For this, we have used homology modeling. We generate the models and fill these loops. We check for the mutational stability, and for that we perform MD simulation runs, and you can see that this model is stable for 100 nanoseconds. We take this stable structure and build a hexameric state of it as well as the monomeric state. Now to know whether this increase in the states are restricting this mutational capacity, we carried out virtual saturation mutagenesis. In this, each residue position is mutated into rest of other 19 residues. For example, in the residue 1, if you have a cysteine, this cysteine is mutated into rest of 19 amino acids. The effect of these substitutions is predicted by Foldex, and according to that, it is classified into stabilizing or destabilizing mutation. Average destabilizing mutation is classified as 0, wherever neutral and stabilizing mutation were termed as 1. We aggregated the score and it gave us a output which is called as mutational capacity, where score 0 represent each and every substitution is destabilizing in nature, wherever score more than 0 were represent as such substitutions are stable or neutral in nature. Residues with mutational capacity 0 were termed as virtually conserved regions, wherever residues with mutational capacity more than 0 were called as virtual variable region. So now the next question is always obvious for a computational background people, that whether our prediction are matching with the experiments or not. So for this, we carried out extensive literature survey, and we found that the already known structurally integrated important positions are also having the mutational capacity score of 0. Wherever in the case of functionally important residue, this score is ranged from 0 till 90, which is expected as the conservation of these residue position are with respect to its importance in the function. So one can maintain the conservation, sequence conservation, despite having a variable or virtual variable position. So with this, we check the distribution of virtually conserved position across different state of dengue protein. I am just showing only for dengue 2 serotypes. I have checked for dengue 1, 3, and 4 also. So here you can clearly see that there is a gradual increase in the number of virtually conserved position from monomer till hexamer. Also, once you can see that the positions which are conserved in the monomer are different from dimer and hexamer. So this suggested that this state change or formation of dimer and hexamer are creating a global level of changes which result in formation of such sites. So from this, it is clear that NS1 structural organization is restricting the mutational capacity, and then it could be important for the evolution. So next, we ask the same question: what could be the effect of this increased virtually conserved region on NS1 conservation? For this, firstly, we identified what are the high or low frequency mutation region in NS1 protein. We take out all the complete viral protein sequences from by NCBI and Viper. We extracted for dengue2, dengue NS1 proteins. Uh, all these extracted proteins were checked for the uniqueness, and then multi-sequential alignment was generated. To know the extent of variability, we perform Shannon entropy scores or Shannon entropy calculations, which range from 0 till 4. 0 represent the full conservation, where 4 represent the presence of each substitution in the equal amount. Now, to categorize the region into high and low SC regions, we check for the distribution of Shannon entropy scores, and you can clearly see that majority of position is highly conserved, and few of them are accumulating mutations. These regions were called as high SC regions, means observed variable regions, wherever this portion is called as low SC, means highly conserved in nature. We also represent this in the dengue NS1 structure. So now, if the virtual conservation is playing some role, mutation capacity is playing some role in NS1 evolution, then what would expect that this virtually conserved region should be of low SC, means sequentially conserved, and vice versa. So for this, we check the distribution of virtually conserved versus virtually variable in high SC regions, and we found that majority of highly variable regions are also virtually variable. This shows that indeed this virtual conservation is playing some role in the NS1 evolution. Now, are we restricted to NS1 of dengue viruses? So for that, we check for NS1 of other viruses like Zika, Japanese encephalitis viruses, West Nile viruses, and we found a similar trend. So this suggests that the quaternary arrangement, which is our main or initial hypothesis, that NS1 affects its protein evolution by increasing the number of virtual conserved sites, which results in restricting overall mutation spread. Now, but we see an interesting observation, and which is that there is a presence of destabilizing mutation in observed variable regions. Now, how a destabilizing mutation can be selected in a viral population? So this led us to ask that there can be other factors which is playing a role in shaping this conservation of NS1 protein, other than the increasing in the virtual conserved positions. So for this, we isolated each mutations and check for its effect on NS1 structures. We see that major of single mutations are destabilizing in nature, as you can see from monomer dimethyl hexamer. Now, this suggested that NS1 does not like to mutate much. Now, what could be this distribution in low versus high SC region? You can see from this graph that the frequency of structurally advantageous mutation, stabilizing or neutral, are higher in high SC region as compared to the low SC, and they are significant with respect to their odd ratios. Now, again, but this is not giving us the answer why the destabilizing mutation is observing in the viral population. Past studies suggested that such observation can be happen, or one can observe such observations if there is some compensatory mutations nearby. So, is this emergence of mutation is due to presence of some compensatory mutation, or compensatory effect by the whole combination of mutation? For this, we visit back our full sequences, and now instead of taking the full sequences, we take the sequences with only the metadata, which we know they are 100% coming from the human patients. We cluster all the sequence with respect to their 100 sequence similarity, and then classified based on their frequencies. These combinations of mutation were extracted from these regions, and we performed the fold-X to check the effect of this combination on NS1 structure. From this, where x-axis represent the high, medium, or the type of cluster based on their frequency, and y-axis represent the percentage of combination. Here you can clearly see that majority of the combination are stabilizing in the functional units, that is dimer and hexamer. Second, you can clearly see that there is a NS1 state-dependent mutational outcome, where majority of the mutation monomer are destabilizing in nature, where they are slowly and gradually turned into stabilizing in both dimer and hexamer. So this hinted toward why a destabilizing mutation can be occur in the population. To further go for its detail, we uh, carried out a detailed analysis on two such mutations, combination 165 and combination 167, where this combination 165 is highly stabilizing in monomer and become highly stabilizing in dimer also. Wherever this combination 167 was highly destabilizing in monomer and after dimerization become highly stabilizing. So for this, we carried out memory simulation runs for in triplicate, and we see this behavior in their RMSD also. We see that a combination 167, which is destabilizing monomer, has not been stabilized for 100 nanoseconds with respect to their RMSD. Wherever in the case of dimer, both the combination are stabilizing and way below than the RMSD of the wild type. So this suggested that these kind of destabilizing mutations can occur due to the stabilizing effect of a whole combination of mutation. Similarly, we came across a pure compensatory pair where L248F mutation is destabilizing in nature but gets stabilized in a combination due to the presence of other mutations such as N223S. Okay. So from this, we come across a model which is depicting the possible regions contributing in the high conservation of NS1 protein. First is reluctance of NS1 to accommodate mutation, as majority of single mutation are destabilizing in nature. Second, this increase in the presence of virtually conserved mutations and their effect in shaping the NS1 landscape. Third, if majority of mutations is destabilizing in nature, then to emerge in the population, you need to have a compensatory mutation. This can be a rate limiting effect, and hence NS1 is highly conserved in nature. Finally, which with to time constraint, I have not shown in the this talk, but we also found that there's none of the mutation, like as you can see here, okay, are present in the dimer or hexamer atrophies. Now, the main thing about this analysis, other than giving a basic architecture or explaining the basics behind the NS1 conservation, is implication of this analysis on identifying of critical or novel interaction as well as druggable sites. 24 unique residues which are present in all the conformation, which are virtually conserved, can be tested for their importance in the structure. Just as we have shown that our virtually conserved positions are also structurally important due to literature. Second, this kind of analysis can also be used to predict a novel interaction site. Okay, a residue is conserved if it is playing a role in structure or function. Okay, so we can do such kind of sequence alignment and look for literature survey, where we have found in the case of NS1 there that the series of proline residues are conserved across the periphery. Proline residues are known to interact with SS3 domain proteins, which are very important and upregulated in the case of dengue or flavivirus infection. So we use that information, perform blind docking to see whether they will interact or not, carried out multiple parameters and see indeed some of the proteins are interacting. We also take the negative controls and confirm this. So this kind of approach can be used to predict the novel interaction sites. Main thing which is used in this can be used from the study is identification of druggable sites. Druggable sites are mainly are used based on their functional importance, but with our study, one can use incorporate the structural significance into picking these druggable sites. Means functional importance plus structural importance give the two more make this site more protective in the sense of getting mutated due to the evolution. So we perform these things. We look for a site which is known for functional importance. We see whether there is a structural important region like lysines are there or not. We also added one more residue which is mutationally conserved in the one virtually conserved, and then finally carry out a vessel screening pipeline, which is published in our previous paper. And finally, this paper is published in 2023 only in ME, And I would like to thank my guide, Professor Sudhavini Sudhir Krishna, my TCM member Anmol Chandele, and uh, a Deepak Ashe, a evolutionary scientist in NCBS. With this, I would also like to thank NCBST for ISCB, my labs, as well as my friend. Thank you.